today. Our speaker is Wesley Culberson. He's an assistant professor in the Department of Medical Physics at the University of Wisconsin-Madison. He's also the director of the University of Wisconsin Accredited Dosimetry Calibration Laboratory. He has a PhD in medical physics, also from the University of Wisconsin-Madison. He's ABR certified in therapeutic radiological physics. Among the other duties he has in his career, he is currently the chair of the AAPM Task Group 292 on Electronic Brachytherapy Dosimetry and the president of the Council on Ionizing Radiation Measurements and Standards. And I know Wesley, he was on my PhD committee, so that's my contact point with him. And without any more talking, I'll let you get into your presentation.
Okay, thank you. Thanks for the introduction. Yeah, this is really nice for me to be able to do this today. I'm going to start sharing my screen. I just clicked to share. Let me get going here. Quick, make sure you guys can see my slide and not presenter view. Is that right? Okay, Maria's nodding. Good. Yeah, this is really, it's an honor to be invited to talk today, and I'm excited because this is kind of a chance to talk about something, not necessarily a full lecture on a physics topic and not a seminar on electronic brachytherapy, which is what I usually talk about with Maria and some other colleagues. And today I'm going to sort of keep it a little more broad, but also talking mostly about calibrations and calibration laboratories, but what's new with the calibration laboratories and the directions that we need to go to evolve with the technology. Got a picture of an old irradiator, and then on the right is a picture of a Varian TrueBeam. This happens to be the one that's in our lab, and we just finished installing it about 2 months ago. We're really excited about it. And it's a pretty unique installation in that it's not used to treat patients. It's just for research and education. So we're really proud of it. But the reason I put it on this slide is that the definition of dose really hasn't changed at all since that Theratron 1000 Curie cobalt unit on the left as it has on the right. And in our calibration labs, in the primary labs and the secondary labs, and everybody involved with trying to determine dose, part of the beauty of our lab is that we don't change. That's what we don't want to do. And so that's why calibrations and calibration labs tends to be a little bit of a dry topic sometimes. But really, we do have to adapt. And in fact, there's a lot going on these days that we have to adapt to. So that's what I'm going to talk a little bit about today. I'm going to start off and make sure I can get through my slides and toggle here. Okay, so thank you. You introduced me, Peter, but I'm going to introduce a little more of myself. This is a personal introduction. Here we are in Wisconsin. As we were starting today, we were talking about it's pretty cold here this winter. We had a pretty mild winter. Now it's a cold snap. These are my two children, 5 and 7 years old. And this is in our front yard where we like to freeze 5 or 6 of these kind of plastic buckets used to store shoes in and put food coloring in and let them sit out for a night. And it's been cold enough this year, we've been able to build the castle. It's actually quite a bit higher than this. It hadn't been above freezing in a while. And then the top picture is kids are learning to ice skate a little bit. This is a lake where some neighbors just went out and shoveled some paths in it, and it was a lot of fun. So we've been having a lot of fun this winter in Wisconsin. It can get cold here like it is where you are, and with having kids though, it makes it a lot more enjoyable. I think the allure is wearing off just a little bit this winter, as it always does coming into March, but that's the way it goes. And then introducing the fact that I have a little bit of a unique pathway into academia. I started as a board certified physicist, actually didn't really intend to end up in the university setting full-time as a academic, but here I am. So I have my board certification and I worked 7 years without any research projects going on at all. I just wanted to learn how to commission machines. I worked in California and Milwaukee, a lot of small field commissioning, CyberKnife and things like that. And then I got the itch to come back to academia. So now I'm in a laboratory. I still have some radiotherapy physics duties that I do for consulting. So I'm still board certified. And again, that kind of makes me a little unique in our department. Our department, we really have to get grants to survive. And I bring in a little more clinical component to that. So that's my niche in the department, is a little clinical perspective. I have 9 PhD students that I'm supervising right now. It's quite a lot, but our lab, I work with another faculty member, Professor DeWord, and we sort of tag team, and we have about a dozen students total. And the senior students are able to mentor the younger students, so it works out quite nicely that I'm not having to answer every question from every student, but it's a fun lab to be in because we have a lot of projects going on. I also lecture for our department graduate program, and as Peter mentioned, I'm the director of the UWADCL. So I'm going to talk about what an ADCL is today. And I know we potentially have a diverse audience here. I was told that mostly people related to the medical physics department at McGill, but there could be dosimetrists here, physicians, and anyone else in the radiation therapy department. So I'm going to kind of introduce what these calibration labs are about, and then also dig into some of the specifics and show some of the setups that we have. Like I said, I was happy to talk today because I feel like there's a connection between University of Wisconsin and McGill. It's been there for a long time before I even got involved. I remember when I was applying for graduate programs in medical physics, there was a very short list of programs in 2001 that offered medical physics graduate programs, and McGill and Wisconsin were at the top of the list. I think the list was about 6 programs or something. And then now it is a ginormous list of, 50 programs at least. And McGill and Wisconsin, I think, are still near the top. So we just have good, strong roots. Our goals are very common, that we want to educate and do research. We have some collaborative projects on electronic brachytherapy. I've been on Peter's committee and joined a research meeting last month, and I thoroughly enjoy that. So let's get started for today. I'm going to separate this talk into about 5 sections and start off with just a quick few slides review of some of the basics, and then move into some of the acronyms that tend to be involved in calibration labs, PSDLs, ADCLs, SSDLs. And then I'm going to talk about our lab in particular, what we do, and some projects that we have underway currently, and then also some directions that we're trying to move in the future. So the big motivation, just like everyone else in radiation therapy that's probably on the call today, is really trying to treat people better, more accurately. And in the world of calibrations, it's more consistently. So we want to be able to standardize the dose between all centers, and also make sure the dose within a center is stable with time, accurate with time, over decades, right? Because these trials and physicians needing to prescribe doses need to be able to rely on that historical data. And if there's these big uncertainties, it just really doesn't help. It really messes up the consistency there. It's also in diagnostic radiology more now than it was before, needing dose more than just a good image. There are laws, I know, in many US states where CT exams have to put the dose on the screen now. That's not trivial to calculate the dose from a live diagnostic exam. So this is growing. It's not going away, needing to realize dose accurately. And we also need to adapt as technologies grow, but we have to make sure these fundamental quantities don't change. All right, so part of us has to stay the same, and part of us needs to grow. Generally in therapy, we want to keep the uncertainties well within that 5% therapeutic window. There's certainly a lot more flexibility in the diagnostic realm. And even within therapy, I'd say brachytherapy has a bigger window. But generally, we want to keep those uncertainties in the 0.5 to 1% range. I mean, that is very small. And that needs to be standardized throughout the whole world, ideally, and definitely within each country and their primary labs. Okay, so there's about 4 slides that I'm copying from Professor Sunchen's from 2009 summer school. All right, maybe I changed the colors a little bit, but I like the way you laid out some of this, just basic concepts. Traceability is what a calibration lab is about, trying to establish a chain that links measurements from a quantity to a standard. And a standard can take on a number of forms. A standard can be an ion chamber, it can be a calorimeter, a standard could be a radioactive source. But it's linking everything together is what traceability is. You can have indirect traceability, you can have very direct traceability, and there's nuances there. So if you have a thermometer and you paid a couple dollars for it, you might have gotten that from a manufacturer that somehow compared that with another thermometer that had a NIST traceable calibration on it, and then that thermometer was maybe compared with maybe a third one, and then maybe it's like 4 steps removed from from one of the primary labs like NIST or the NRC in Canada. So that's very indirect traceability. And we want to keep things as direct as possible. That calibration path is needs to be as small and as tight as it can be. So just giving one graphical example of traceability, if you have absorbed dose to water, this is one of the most common therapy calibrations from a primary standards lab. I have NIST in parentheses. The NRC is an example of a primary lab. And a water tank, you're gonna have a cobalt radiator that is usually going straight down into this tank, and then in there you're gonna put a calorimeter. So that little orange rectangle there with the two white lines in it is two thermistors that are measuring the temperature rise in a water tank. Once you get the temperature rise, you can convert that to dose through the specific heat of the material. And then once you have dose, then you can take an ordinary ion chamber and put it in the beam in the same spot. Generally, this needs to be done right after one after another. And that ion chamber then gets a reading. And so now you have the dose per reading on that ion chamber. And that ion chamber is much more portable than a calorimeter. Calorimeters are very difficult to take a measurement, very time consuming, very finicky. So they aren't usually done very often. But these ion chambers, then you can just put it in the mail and send it on to another lab. And that's like an ADCL where I work, an SSDL. Then we could take that chamber with the calibration on it from a primary lab, put it in the beam and get a reading and convert that to dose. And once we know the dose right there, then immediately after we can put in a physicist chamber from a hospital or a customer, any other customer. And then now we can provide a calibration for that chamber. That's called NDW for the TG51 protocol. That NDW then, once it's established, we'll put it on a little sticker, put it on the box, and send that to a hospital where they can then put it in their beam. But that's no cobalt anymore. It's going to be a linear accelerator. So now they have that NDW calibration. They need to apply an offset for the beam being slightly different, KQ factor, and then they can determine the dose themselves. So NDW is ultimately traceable to the primary quantity, which is dose, established with a standard, which is the calorimeter at a primary lab. Okay, so that's the lingo. Now, each one of these three steps along the way has some watchdogs in place to keep them accountable. The BIPM is an international organization that will send chambers around to all the primary labs and make comparisons. Then the AAPM has a subcommittee that reports straight up the chain to the executive committee called the CLA, Calibration Lab Accreditation Subcommittee. And then each clinic then can use a service such as IROC Houston. Other places do this as well, where you can get an independent measurement of your dose. So those are the watchdogs that are put in place at each step along that traceability chain. Okay, so now let's talk a little more about these acronyms. A PSDL, Primary Standards Dosimetry Lab. This can be the NRC. That's not too far from you guys. It's NIST in the United States. It's a national lab usually set up by the government. And that's for the purpose of developing and maintaining these primary standards like that calorimeter. All right, no clinic needs to have a calorimeter. That's the purpose here. There's one lab and the government really keeps track of those standards. And in some countries, there aren't PSDLs though. There's a lot of countries in the world. They can't all have PSDLs. There's not enough resources. So in those countries, then they're gonna have secondary labs. In these primary labs, those standards are using what are called absolute dosimeters. And absolute dosimetry is measuring dose using something that doesn't need a calibration. A calorimeter doesn't need a calibration. It's a primary measurement. You take the temperature change and you multiply it times the specific heat, gives you dose. You don't need to send that to another lab. But in a clinical context, you don't ever deal with primary standards. You're going to deal with clinical reference dosimetry. Now, someday that might change with miniaturized calorimeters, that's for sure. And Jan can tell you all about those. But for now, right now, everything is a secondary reference dosimeter. A little side note on this. This tends to come up and there's some confusion because of the way the manufacturers have written their software. So if any of you guys have one of these fancy Sun Nuclear ARC checks or map checks and you put it in the beam, it's got a little file drop-down menu that says absolute calibration. It's always kind of bothered me a little bit, because I know, and I was in a cal lab when I was a grad student, that that's not actually an absolute calibration at all. So the lingo gets a little confusing, but it's often denoted, that terminology in a clinic is often used just to kind of distinguish between a relative measurement and a dose-based measurement. Okay, a dose-based calibration is what that is. And they use the term absolute, even though it's really not an absolute measurement. So if metrology experts, we use that wording a little differently. Just wanted to clarify that if you're ever asked that in the board exams or anything, there is definitely a big difference between an absolute calibration on a map check than there is at NIST or the NRC. Okay, there's all kinds of organizations, and I was lining them up for this talk, and I realized they're all 4-letter acronyms. BAPM, the ICRU, the IAEA, SSDLs, ADCLs, NIST, NRC, C, usually is the 4 letters there. And why all these acronyms? Well, that's just how it is. And the BAPM I mentioned is one that is a very old one, established by the Meter Convention, well over 100 years ago. The Ionizing Radiation Division, really, they're the ones who run these comparisons between the primary labs, and I think of them as kind of the top of the food chain. This is the data that I got from Ronnie Manides. He is a physicist at NIST, showing a comparison from a few years ago. This is absorbed dose to water. I'm sorry, this is a air kerma from cobalt-60, and you can see all the values from around the world. You can see the NRC down there about halfway down the row, and NIST is there. All of them are within 0.5%. That's the goal. 0.5, 0.6% are sort of the limits on that intercomparison. It's really impressive that they can all compare so well, and that's the goal from these primary measurements. There's also another organization that comes into play called the ICRU. They define quantities and units, and they actually just came out with a report a couple years ago, ICRU 90, where some of the fundamental quantities of dose are actually more related to air kerma have changed. The ionization potential, I, for some of you that have learned the nuances of air kerma, changed its value enough that all of the standards in the world are going to be changing by about 0.5% for air kerma, for cobalt, from cesium-137. And that's not a lot, but it's definitely significant in our world, and it's being implemented very slowly in the US. We haven't implemented those yet. I'm not sure if NRC has or not. And so, but what happens when something like that happens and a new unit is defined with a new quantity, then it has to be disseminated. It takes a lot of effort, and that's what the ICRU works on. Then there's the IAEA network. The IAEA deals with calibration labs. It consists of the primary labs, SSDLs, which are secondary labs, and they have this exotic lab network that goes into a lot of underrepresented countries in the world, and they do a really nice job of keeping all these labs standardized. They do intercomparisons, not as frequently as maybe some would like, but they really are very pertinent for calibrations, and Canada is part of this SSDL network as well, and the NRC is involved. This is a map of the whole world showing all the different affiliate members. The United States is a little odd in that we only have the primary lab affiliation. We don't have any SSDLs at all. Okay, so then what's an ADCL? So my title was director of the UWADCL. This is an accredited lab. It's very similar to a SSDL in the IAEA network, but it's a little different. One is that we have an accreditation from the AAPM, okay, not the IAEA. And in that accreditation, we have a few different standards that are imposed on us. We have different criteria. We have to do a lot more intercomparisons, and we get auditors from the AAPM on site every 2 years, and that distinguishes us from IAEA labs. Okay, there are 3 in the United States right now. There's been more than that over the years, but right now there's just 3. 2 are part of universities like ours and MD Anderson, and then KNS is a private lab in Nashville, Tennessee. And we provide users with calibrations. And like I said, we provide everything an SSDL would, but we also have extra proficiency tests, meaning extra round robin tests with equipment. And another big thing that distinguishes ADCLs is that we are not allowed to be affiliated with instrument manufacturers. And so it removes some of the potential bias. And that's different. A lot of SSDLs in the world happen to be with manufacturers, and it is very convenient, right? If you buy an ion chamber from a manufacturer, and if they have a cal lab, then it's very nice to just have them calibrate that and have faith in that number. But in the United States, and actually North America overall, in Canada as well, they're not affiliated with manufacturers. And I really like that, because then there's no bias on what equipment, when they recommend equipment, and any other bias at all. Okay, what does an ADCL not do? Well, we calibrate a lot of things, but we don't calibrate solid state detectors. And this is a surprise to some people, because there are so many diodes in use in radiation therapy these days. Diodes, arrays, there's diodes for diagnostic systems that measure dose and have value. I mean, there's diodes everywhere. They are a great tool to have in the toolbox, and I highly recommend them as well. But their calibrations, you can't get them calibrated by an ADCL. And the reason why, I think I'm going to have that on the next slide. But basically, these are a list of things we don't calibrate. We don't calibrate TLDs. We don't calibrate scintillators. We don't calibrate diamond detectors, OSLDs, or any form of solid state diagnostic instrument system. We also don't calibrate pulsed electrometer scales or AC current electrometer scales. And in North America, we don't calibrate systems for therapy. We don't, the ADCLs always separate the ion chamber from the electrometer. Okay, and that's not the case around the world. So if you go to Europe, you're easily going to you can get a system calibration. I think it's a challenge when you have an ion chamber married with an electrometer, because that calibration only holds for that system. So if you need to swap anything out, you can't do that. So I really like this independent system that we have. Okay, why not a diode? This question comes up fairly often. It's ultimately not a reference dosimeter defined by TG51, but what does that mean? Well, basically, if you were to calibrate a diode today in front of that cobalt beam I showed you, and we want a 0.5% uncertainty on it, and then you ship it back to a clinical physicist, and they take a measurement the next week and the room temperature is different, that diode calibration is going to not hold. You're going to get a different response because solid state devices are temperature dependent. They're also very energy dependent. So if you calibrate it on a cobalt beam, you need to go to a LINAC beam, and there's not a list of KQ factors for diodes. Now you could come up with some, but since they're so sensitive to energy, then they're not going to hold with nearly the accuracy that's needed. So then why are we using diodes? It's because they're really sensitive, very small, excellent relative dosimeters. So these arrays like a map check or an arc check or a delta 4 are great arrays with over 4,000 diodes at once, because at once you can map out a dose distribution very well. And you can do that day after day, as long as it's mostly a relative measurement. Now you will also find that a lot of these phantom manufacturers with the diodes in them have software that is able to account for those temperatures very well. Okay, so you'll find that you can calibrate it on a Tuesday night when you do your QA, and you could come back for a month and you're probably okay with that calibration, even if the room temperature is different, because they have internal temperature probes that are able to adjust those readings. And they have it worked out really well. It's just not something that a calibration lab can do. All right, so there's a distinction there. And that's why you can't send it to us and we can't offer a calibration on it. Okay, what about Canada? I know this talk is fairly biased towards the US because that's where I'm from and that's the system I'm most familiar with, but we tend to have a lot of synergy in North America overall. Canadian physicists send their equipment directly to the NRC. So you don't have ADCLs, and as far as I know, not to my knowledge and not to Brian Muir's knowledge, who I talked to yesterday, you don't have SSDLs. And it's just because there's fewer clinics in Canada. They're mostly large centers. NRC can keep up with the demand. Brian mentioned yesterday that they calibrate about 60 chambers for Cobalt 60 each year. And then I think about a quarter or a third of those are calibrated on a LINAC as well. And there are no SSDLs in Canada, although, you know, that slide I showed you earlier, I think that NRC is affiliated as an SSDL in some ways. But anyways, this is sort of the slight difference in Canada and the US. So the UWADCL in the US, since we do so many more calibrations, NIST, our national lab, cannot handle that amount of work, essentially, is what it comes down to. So many years ago, ADCLs were set up to basically release that strain on our national lab. So we're a secondary lab network in the US. Our particular lab at the University of Wisconsin, we're of course the biggest and the best, and we're really modest as well, and humble. We've been around a long time, way before my time. And Professor DeWord founded the lab. He's worked as director for many years, and he gave the reins over to me about 5 years ago, but he's still involved, still does some research in the lab. And I think a lot of you guys a lot of people might know him by name at least. And now the ADCLs and part of what we call the UWMRRC. So the ADCL, it's self-accredited by the AAPM, is not accredited to do any research. So we're just part of a bigger dosimetry lab that we call UW-MERC, and that has TLDs. We do research projects. We do other calibrations that aren't accredited by the AAPM, such as survey meters, like a Geiger counter. We'll calibrate that. And we are also A2LA accredited. And basically our mission is a bit more broad. We want to educate and calibrate and do research. So I'm going to talk a little more about what we do for calibrations and then what we do for research. So don't try to read all the screen, but basically these are the areas that are under our AAPM scope. We do ion chambers, we do diagnostic chambers. We will do diagnostic systems, just not therapy systems. And we do lots of brachytherapy calibrations. We do lots of brachytherapy measurements of sources as well. And we'll calibrate electrometers, both analog or digital. This is just sort of a random and interesting graph I'm showing over the last 10 years. This is a number of cobalt-60 ion chambers that we've calibrated each year. And you can see, remember I told you earlier, the NRC does about 60 per year. In our lab, we do about 2,000 per year. So there's someone down the hall right now doing about 10 or 15 today. That's about how many we do per day-ish. And it has a cycle. It's a really funny cycle that when I started, it just keeps going. Every year we get this funny cycle, and we've always tried to figure it out. And I think the best guess that we have is that when the reference dosimetry protocol switched from TG21 to TG51, all the clinical physicists in the country just sent their chambers in for a new calibration that year. And then since there's a 2-year recommendation on calibrations, that cycle has stuck, and it goes up and down every 2 years. And so that's just kind of something interesting. Another thing to see here is that dose to water calibrations continue to increase. That's ADW on this graph, and air kerma calibrations decrease. Okay, this is our cobalt calibration setup. This looks different than it would look in a clinic, and the reason is that we have different variables we need to try to deal with. One is you can see these resin beads floating. It's a really neat idea. It keeps evaporation down, and that serves two purposes. One is just practically speaking, we don't have to fill the tank up as often. And the second thing is that it reduces our evaporative cooling. This is something that I never noticed when I was in the clinic, but if you measure the temperature of your water with a very large surface to a very dry air, perhaps, it's actually going to be 1 to 2 degrees cooler than the air temperature surrounding it. And it usually, your chamber will come into equilibrium pretty quickly. It only takes a few minutes, maybe 5 minutes, and you never even really notice it because you're setting up a tank anyways. But in a cal lab, when we do 10 or 15 calibrations per morning, we need every minute we can get. So with those beads there, it drastically reduces the evaporative cooling. And now the temperature between the shelves in the corner of the room and the water in the tank is only about a half a degree or less. And so that means it only takes a very short amount of time to come to equilibrium. We utilize plumb bob lines on the sides to line the chambers up. And it's a very accurate way to do that. It's a replacement technique, which means these chambers are put about 5 centimeters deep. It doesn't matter if it's exactly 5 centimeters. What really matters is that you put that NIST chamber in there in the same spot that the customer's chamber is going to go in. It's called a replacement technique. And like I said, it doesn't matter if there's a little window bow, if we're 5 1/4 centimeters or 5 centimeters, because the calibration holds with depth. It just doesn't hold when you change depths between the two chambers you're dealing with. Okay, these calibrations, that's the tank. It's shown zoomed out a little bit. This is an older version of our tank. We now have full automation on the top to position the chamber with the light behind the plumb bobs there, you can see. And then this is a cobalt-60 irradiator in the background. So electrometer calibrations, the main goal here is to inject a very known charge into each electrometer. So we have these 4 stations in the lab where we can do electrometer calibrations. They take a long time, actually. And we have NIST traceable resistors, a NIST traceable voltmeter, and a timer that we can use for charge calibration, and then a NIST traceable current source for current calibrations. We do not do pulsed mode calibrations. There is no NIST standard for a pulsed beam, and I'm going to have a little picture on that later. And you might ask yourself, wait, my electrometer has something called pulsed mode on it. This is the PC electrometer from Sun Nuclear. I'm not picking on them. They just happen to have a software toggle between continuous mode and pulse mode. But every digital electrometer has to have a way to deal with a pulse beam. So when you have 2 modes on your electrometer, we can't calibrate it in pulse mode in a calibration lab. They have the same circuitry on the inside, but the software deals with them differently. One question we get is, can anyone do this? And the answer is no, not at least in North America. No one has the ability to calibrate a pulse beam. Remember, a LINAC is a pulse beam. So this is a very important thing to remember. And just be very cautious if you're using one of these types of electrometers, that if you're using it in pulse mode and you can't get it calibrated in pulse mode, you might want to just keep that confined to relative measurements, scanning PDDs and things like that. Okay, now recall just a little, this is from Karsmark. We have 2 different types of pulses in our beams. We've got a really high frequency pulse, line C, that's 3,000 megahertz. That's the microwave, klystron, magnetron. And then we also have a little bigger pulse, which are the pulses that really affect the ion chamber and the electrometer. Those are about 5 microseconds separated by 5 milliseconds, so a very short duty cycle. And then in the world of diagnostic imaging, you also have some sort of pulses, but they're not really big enough to affect an electrometer. So you're safe to use those pulse mode or continuous mode electrometers for diagnostic sources and for brachytherapy, because brachytherapy are constant sources as well. So speaking of brachytherapy, this is what our calibration workstation looks like. Not very complicated. We have a pressurized well chamber that we use here in the lab, and then we will be able to calibrate pressurized and vented chambers for low and high dose rate sources. We measure seeds as well, generally just for the seed manufacturers, but sometimes a very busy center doesn't want to let their well chamber leave, so they'll get a source ordered, send it to a cal lab. We can measure the air current strength and then send it on to a hospital. And then you can do the cross calibration yourself. That's a very viable way to calibrate your well chamber. This shows our X-ray tubes. We have a couple of them in our lab. We have a bunch of different filter packs. We can make a whole laundry list of different beam qualities, mostly diagnostic, but also for orthovoltage therapy treatments and things like that. We don't in our lab currently have an accredited dose to water calibration. I think there is at the NRC. We're working towards that. It's just not NIST. NIST doesn't have that yet, so we can't be traceable to them. Just a big picture note on what are our sources of variation. I mean, mainly it's the small thermal fluctuations in ion chambers that change during a calibration. Otherwise, we're really tight when we do a measurement. It's mostly the end user that has variations that they have to think about. First of all, your detector will drift with time. Okay, sometimes not by much, but it's your drift after it gets calibrated that you have to keep an eye on, and generally you're recommended you send it back every couple of years. Other considerations are waterproofing. We just had one in our lab this week. We thought, hey, this has a little crack in it. And we put it in our water tank, and the little graphite on the inside got dark. And so we knew a little water was seeping in there, but it wasn't enough that the customer actually knew it was happening. And so you really want to take special care of these waterproof chambers, try not to bang them around. And then also make sure those dust caps are on them, because a little dust in there gets across the insulator and it adds a little current to the reading, and you don't want that. Your calibration will change with just your triax connection there. There's issues with altitude effects. I'll talk about that in a little bit. Most of the time it's not an issue, but well chambers, it is an issue. This is the difference in a well chamber calibration with just a palladium seed. If you're in Denver or Mexico City, you're going to get a 10 to 20% different reading. And that's because the dimensions of the cavity in a well chamber are on the same order of magnitude of the range of those electrons that are set in motion. So if you change the pressure or the mass of the gas in there, you change the calibration totally. It doesn't just go by the PV equals nRT law anymore. So that's something important to keep in mind. Another thing in general with calibration labs, I always like to say this, it's not just a number. It's very easy as a clinical physicist to take that number, put it in the file cabinet, slap a sticker on your ion chamber and move on. But really, it's not our job necessarily as a cal lab to keep track of your equipment with time. So when you get that number back in your lab, why don't you look at it and compare it to the previous one and the one before that? That is very important, especially if you send your equipment to maybe a couple of different labs. The labs themselves aren't going to pick up on it. So it's your job to do that. That number has meaning. Look for outliers, look for something that doesn't make sense, like a sudden jump or something like that. And also another note, calibration labs aren't meant to be repair shops. That's what the manufacturers are for. Okay. Just a side note on manufacturer calibrations, they offer calibrations and sometimes they're very accurate, but they don't have the same system in place as other network like the SSDL and the ADCL network. So there can be some issues. They don't have the round robin testing that's required. So I would use those values with caution, a manufacturer calibration. Okay, a couple things, moving on to just our lab. We're very fortunate, we have a free air chamber, and this was developed here in the 80s, and now it's in a few labs around the world, and it's in the textbook by ADDX, if any of you have that. That's our ADDX free air chamber to measure air kerma. The first serial number 1 well chamber, HDR 1000, was developed in the lab, and we still use serial number 1 to do HDR calibrations. We also have developed the air kerma standard for HDR. And then this is a project of mine when I was a grad student. We have a very large free air chamber for brachytherapy seeds. And this one's unique in that it has variable apertures. And we've used it to establish standards for several new low dose rate brachytherapy sources. So we're very proud of that. We are also the only lab in North America that will calibrate for electronic brachytherapy, a well chamber with NIST traceability. So a special insert was developed for that HDR 1000 plus chamber, shown in the bottom right there. And then this is what it looks like when it's removed from a well chamber. And so now I'm going to move into some of the projects that we have underway. One is to measure the TG43 parameters for the electronic brachytherapy source, the Zoft brand name. This is the Zoft accent source. We've been working on this for a long time, and the publication's pending. It required a lot of TLD measurements and a lot of Monte Carlo calculations with different codes to sort out some of those cross-section issues in the codes for these low energies. We also are looking at the relative biological effectiveness of electronic brachytherapy versus iridium-192. This is a very important topic that hasn't been flushed out yet. It's also a very difficult project to do. It's hard enough to determine what the dose is from electronic brachytherapy. It's even harder to try to culture cells and look at their survival curves. So what we have is a number of different Petri dish styles with dosimeters that we can put in them. TLDs are shown in this picture. We can do an iridium irradiation followed by a Zoft electronic brachytherapy irradiation and look at if those cells are dying at the same rate or different rates. And ultimately, especially for gynecological treatments, the RBE has a big role to play. And right now people are using a generic value and it has not been proven. And there's some centers, some hospitals refuse to use electronic brachytherapy at all because RBE just hasn't been determined yet. So this is an ongoing project. It's very fun. It involves us growing a lot of cells down the hallway and trying to irradiate them. And hopefully we'll be making some progress there. I mentioned pulsed beams before. So I'm interested, I have a graduate student working at looking at the effects of these pulse beams with digital sampling. So what I have here is a whole bunch of rows. This is essentially the pulses from a LINAC. If you guys have a variant LINAC, they generally are sequences of 6 pulses. And what I have mapped out here, the blue line is a farmer chamber, and the green and the red lines up top are the ionization currents from a larger volume chamber. And you can see that it doesn't clear its pulse. It doesn't clear out the ions very quickly at all, and it starts to run into the neighboring pulses. And when it does that, electrometers can't handle that very well. They don't all handle that the same way, and you end up getting different readings. So you can actually have a reading on your electrometer that is incorrect because it was calibrated with a constant current source. But if it gets pulses in a very large ion chamber or a medium-sized chamber, you're gonna get a different reading. It's a bad thing. And fortunately, it doesn't come into play too often, but it's something that we're trying to flush out. We're also looking at more sources for altitude effects, cesium and palladium-103. There's a student here that's measuring how the well chambers are affected by the altitude. Remember Denver, Mexico City kind of thing, but now we have different source models. We looked at establishing the Cividot. The Cividot is a brachytherapy source that is directional. It has a gold shield on one side, and they're shown there in yellow. It's very directional. It's the first commercial directional brachytherapy source, and it needs AAPM data for it. So we were able to measure it on a free air chamber. This is what it's shown when it's incorporated into a sheet. So these sheets are put into surgical cavities, and the membranes just dissolve away. They're bioabsorbable with time, which just leaves these sources. We were able to measure it on a free air chamber, that same free air chamber I showed you earlier. And then we were able to put a special well chamber insert, kind of clamp onto these sources. One thing we discovered along the way is that well chambers are no longer very symmetric with these asymmetric sources. So you have to measure at all 4 angles. This shows the relative reading, changing by a few percent. We also noticed that sometimes the gold shields come off of the sources, and you can only detect that by doing an anisotropy measurement. So this shows the red line here shows one of the gold shields slipped off a little bit, and that's a bad thing because then the dosimetry is going to be way off. So we were able to discover these few things in the lab. And then moving into the final part of this presentation, and by the way, I just noticed these slide numbers at the bottom are not accurate. I didn't update them, so we don't have 20 more slides left, but I am going to mosey on through here so I don't take too much more of your time. We'd like to move towards LINAC-based calibrations. This is something that's old hat for you guys in Canada. The NRC has been doing this a long time, but NIST hasn't done it yet. So hopefully the ADCLs will be able to do LINAC-based calibrations. There's always a discussion involved there on whether or not you need to do that or not. I'm not going to go into that today. There's a good article in the Medical Physics Journal where they debated that. One big push we've been doing the last several years in our lab is to look at these radiobiology X-ray beams. So this is a cabinet irradiator that's used to irradiate mice or cells for preclinical studies. Okay, all radiation trials start with a mouse getting irradiated in one of these irradiators. The X-ray spectra in one of these irradiators doesn't match anything that has primary lab traceability. It's quite a problem. And the people that have these irradiators don't really care, generally speaking. These are biologists and they don't have medical physicists on staff. So we did a survey where we sent out these mouse mimicking phantoms, sent them to 12 different facilities, said, give it 1 gray or give it 4 gray and send it back to us. And when it came back, the differences were up to about 40% on some of the irradiators, with the X-ray irradiators being the worst. And so the issues are that the beams don't match. The different experiments have different scatter conditions. So there's different sized mice. The backscatter isn't accounted for at all, really, not properly at least. And so I've got a student who's looking at something. My favorite part about this project is the name of it, the mouse put factors. And so what he's done is 3D printed these different mice sizes, put them in an irradiator to see if the output factor changes, because all these mice are considered the same for a radiobiology experiment. And so we put TLDs in all of them, and now we've got a graph of mouse size versus dose, essentially, versus timer setting on one of these. So that's a good, fun project. This is the picture I took just yesterday. We have a new, larger free air chamber for medium to high energies. This is one of our students in front of a 500 kV X-ray tube, trying to establish new beams for these X-ray irradiators for radiobiologists to use. And so those new beams should have new pathways of traceability. This shows a 500 X-ray tube. These are brand new. First 500 X-ray tube came out just a year or two ago. So we have one now to do some research on. And we think it could work its way into the medical arena, because why not? It's getting closer to electron therapy, and it's something you can turn on and off and you don't have to shield it as much. So it could work its way in. So we're doing some research with that. This is another picture of the free air chamber that will go in front of that X-ray tube. Another slide here on someday we hope to have absorbed dose to water for X-ray beams. And this is something we're using this tank for. We hope to have NIST traceability someday on this. Flash therapy and TRT dosimetry are the last 2 topics I'm going to talk about. Flash therapy, we're not doing anything different in the calibration lab. This is very high dose rates, but the beam qualities are still the same. There's a lot of groups in the world working on dosimetry for this. And essentially, the conditions are that with those high dose rates, you're going to have recombination, which is an issue. And so what you want to do is have a very fast chamber and overcome that recombination. And the trivia question for all the PhD students here, what makes an ion chamber fast? Well, you typically need small plate separation, a very high electric field, and maybe even use a different gas so that your electrons don't recombine with heavy ions. You want it to be very fast. Okay, so that's the focus of flash therapy. In terms of calibrations, it'll still probably be cobalt-60. TRT dosimetry, this is my last topic here. It takes many forms. Alpha emitters, beta emitters. It can be small areas of the body. It can be very widely spread throughout the body. It gets really messy. So these are liquids. And generally, TRT dosimetry uses a dose calibrator. This is something in nuclear medicine. You have a syringe, you put it in the dose calibrator, you get activity. Okay, there is a calibration. It is NIST traceable, but it's not nearly the precision we're used to and the accuracy we're used to. And we'd really like to be able to connect these radionuclide therapies to dose to water, because that's what the patient's really getting. Otherwise, people are mostly concerned about diagnostic qualities and not so much the dose. So it gets messy though. A lot of Monte Carlo involved. And this is a project we're working on. I'm working on with Siemens. This is a phantom that we fill with these TRT agents, putting a number of different detectors in there. This is a diode that's shown here at the bottom. Its issue is it's angular dependent. We also use TLDs, ion chambers, and we're injecting all these radionuclides in this phantom and trying to correlate it with the Monte Carlo calculations. So that's a lot of groups are working on this, different approaches. That's the end of the scientific portion. I wanted to show you now this last fall, a fun project, because our lab, we like to horse around and have fun. We got rid of our old Varian Linac, and we decided to have a little fun with it. I don't know if you guys have done this up there. It's a lot of fun. It's very hard to get the opportunity to do it, but if you can pull the target out of your accelerator, put it in photon mode, and use the electron beam itself, it scatters a little bit off the mirror and things so that it creates a wide beam. Put a piece of plastic in the beam, and then if you're able to tune it up just right, you can get basically kilograve dose in a few seconds. So we irradiated these plastics, and then when you put a nail on these plastics, you walk in the room and you can kind of hear this sizzling and cracking. I'm not kidding at all. And you wear these insulating gloves, and you put a nail on the plastic and you tap it, and that nail is tied to ground. It'll go boom, it makes this flash of light, and it creates these what are called electron trees. These are essentially all the electrons from that electron beam get stuck in that plastic, and then they short right to that nail, and they do it so violently that they melt the plastic on the way, and it creates these beautiful, beautiful electron trees. And I don't know if you've seen these before. I've seen them. I've never made them before. It's quite exciting. And what you can see are 4 different trees we made with different methods. We irradiated that one on the left from the top and the bottom only. And so 6 MeV electrons, they only go a couple centimeters. So you can see those white parts of that tree is where all the electrons stopped. And then when we shorted it out with the nail on top, they all shorted out the top, and we did the same thing on the other side. And then the other ones were irradiated more uniformly. The one on the right there looks like angel's wings. We hit it from a 6 MeV beam on each side. And then, of course, you got to put a little LED light under all these. They look really pretty. This is one of the trees we made on a big piece of acrylic plastic. Put a little light under it. Quite beautiful. It's quite fun physics. Not a lot to do with medical physics, but it's quite fun. Okay, so in conclusions, the calibration lab network's complex, but it serves us very well in North America. We've got a very tight system, both in Canada and in the US. Equipment calibrations require a lot of consistency. They also require a lot of research and development to stay current and accurate. And that brings me to the end of the talk. I want to acknowledge, got a bunch of students and staff that help with a lot of these projects, and also the people that send their equipment to our lab. Their calibrations help fund the research. So that brings me to the end, and I'm happy to take some questions here.